Good afternoon, everybody. You all know me, so I don't need to introduce myself. So, I welcome you all to these webinar series that have been started for interns, primarily to make the best use of this lockdown period so that you are in touch with your theoretical knowledge. Although the practical knowledge is slightly on the backhand these days, but we will still try to compensate it with these webinar series. And so I think it's a very good initiative. As for Department of Orthodontics, we've decided that we would be taking up topics which would be important for your entrance point of view also. And a few topics we know that there are some concepts that are not that clear to you when you read it in final year, because as it is, you don't get time to read the books that we recommend you. So here I present all the culmination of the latest articles, of the landmark articles, and the book series so that I'm able to clear some of your concepts about the biology of tooth movement, specifically the mediators, and also so that you're able to answer any question that comes from this topic. I would be circulating a quiz after this lecture, so please be sure that the answers of the quiz will be hidden somewhere in the slides only. So be very careful with the slides. And I definitely say that I don't know everything about the subject, although I've been studying this since my thesis days. It's one of my favorite topics, the biological mediators. I keep on brushing my knowledge, I keep on attending, I keep on lecturing, I keep on reading, but still the knowledge is never complete, and that should be our motto, that we should try to gain knowledge from whatever source we can, and it is an unending process. Well, when I had taken a PG seat in ortho, I had definitely thought that I'm going to a clinical branch. And the next thing would be that the next day they would make me enter the clinics and they would tell me how to put the brackets and the wires. And that is what I would be doing for the rest of my life. Definitely, those of you who are interested in doing ortho PG after this, let me tell you, it doesn't happen so soon. We have preclinicals, we have typhos, and quite a lot of time passes before we actually touch a patient. But it's rewarding. It's rewarding to treat patients. So soon I had started putting these brackets, I'd started putting these wires, and obviously I had done all kinds of tooth movement possible, like retraction, expansion, functional movements, orthopedic forces, orthodontic forces, every kind of forces I was putting. But then what also interested me was what was happening underneath. Because I knew that tooth is not a singular object which is placed in the mouth. It is attached to the periodontal ligament, it is attached to the bone, it is having effect on the gingiva, on the pulp. So it is affecting a lot of things. And what is the culmination of all the things that it is affecting? I should be knowing that. So I got interested in what goes underneath when I put any kind of force. There are so many questions that come to your mind. And so I started reading. I would just be putting up the major references that I have read over and over again. This Vinod Krishnan article of 2006 and Masella 2006 is just landmark articles. There are some newer articles also. I'm sure that you will not be having time to read through them. So I have prepared a gist for you all and my understanding I'm presenting here. So this was a kind of a flowchart that was formulated by Mustafa, published in AGODU 1983, which will give you a basic framework of what happens when the orthodontic forces are put on the tooth. We know that there are so many theories that are circulating for the orthodontic tooth movement: pressure tension, bone bending, piezoelectricity, bioelectric theory, and also the inflammatory theory. But do you think that they are all secluded, that they don't work in unison? You're soon to find out, because everything overlaps. It is a single tooth on which a force is applied, and the surrounding all the periodontal structures are getting affected. So but one particular theory is not going to be applicable anywhere. It is an overlap of all these theories that will make you understand what happened when the orthodontic force is applied. So if you see here, in the pathway 1, Mustafa talks about bone bending. Bone bending, definitely when a bone is going to bend, there are going to be produce some electrical signals in the organic as well as the inorganic matrix. The second thing is it would also be leading to formulation of the second messengers, prostaglandins. The second pathway is going to talk about the tissue injury, which is basically inflammatory, and then it is going to release so many mediators and enzymes. Together with the bone bending, the tissue injury, they are going to culminate and lead to orthodontic tooth movement. We are going to go into the detail and understand the process. So this is my favorite part. So what happens? If we just start with the tooth structure or diagram, if I have to explain. So I have on the right side put 4 lines so that you can see that a single orthodontic force is not only affecting the tooth, it is affecting all the structures like gingiva, alveolar bone, pedontal ligament, and the pulp. Individually, how each structure takes on this force is different. One structure which is not given due importance but which has a major role in taking up the brunt of the orthodontic force is the extracellular matrix. You know, cells are not lined on their own. They have their own matrix, a cushion surrounding them. So you talk about any cell, be it an alveolar bone cell, an osteocyte, osteoblast, osteoclast, fibroclast, any kind of cell, it has its own cellular matrix. So whatever tissue injury, whatever injury we put on the tooth, is not directly transmitted to the cells. It is first taken up by the extracellular matrix. Mark each word. Just look at the diagram. So this extracellular matrix is then in touch with the plasma membrane of the cell. Inside you have the cytoplasm, which has a cytoskeletal network. Beneath that you have a nuclear envelope, and in the nucleus you have the genetic structure. So you see, this is at the microscopic level that I'm seeing. But we have to understand this because the extracellular matrix, which is comprising of the collagen, fibronectin, vitronectin, all these are proteins which are present on the outside as a cushion, and they formed an intertwined, entangled network. It's a kind of a cushion. Whenever you put some injury, what happens? Now this cushion is going to get disturbed. Collagen network, fibronectin, vitronectin, this network is going to get disturbed. The extracellular matrix is going to get disturbed. Now it wants to transmit this disturbance inside. How will this disturbance transmit inside? There has to be some connection. This connection is in the form of integrins. Integrins are those proteins which have connect on the outside as well as on the inside. So when you talk about the outside, it is going to connect to the collagen. And when you talk about the inside, it's going to connect to the cytoskeletal network. Cytoskeletal network is a network of proteins which is present in the cytoplasm. They also will have the brunt of that injury which was happening outside. How? Integrins will take that. It will reformulate its protein structure. On the inside, it has talin, one protein, which is again going to attach to the cytoskeletal proteins like microfilaments, intermediate filaments. So all these are going to get disturbed, and once they get disturbed, the protein structure gets changed. It will go to the nuclear envelope and then it will internalize. So basically, this signal gets transmitted towards inside. Once it gets transmitted towards inside and the genetic structure changes, only then the cell decides what I have to do now. Whether I have to do bone desorption, bone formation, or I have to resorb the fibroblasts, any kind of activity that the cell has to do, it's only after the genes tell them what to do. For that, the signal has to go inside. So that's how it goes. If you talk about periodontium, initially when you apply the force on the tooth, what happens? Again, periodontal ligament is like a cushion. So the tooth is going to push it where you apply the pressure on the opposite side. It's going to push towards the alveolar bone. So that periodontal ligament space is going to get reduced. First structure which is going to get affected here are the blood vessels. The blood vessels then, because they line with each other and they will form a precapillary network kind of a structure, then they will form the inflammatory effects like the vasodilation, and they will release the leukocytes in the localized area. That is the time the mediators are going to get released. Let's talk about alveolar bone. Now this is very, very important because all we are dealing it is, when we talk about the bone metabolism, is bone resorption and bone formation. So how this happens? When you put the pressure on the bone, one osteoblast is there. It is lying very silent, very dormant. It doesn't know what to do. What it has on its structure are the TNF receptor, which is the tumor necrosis factor receptor, which are present on the osteoblast. Another form of receptors which are present are interleukin 1 beta receptors. So initially, when the force is put on the tooth, like we've explained in the periodontal ligament, that the precapillary network is formed, there is vasodilation and the leukocytes are released and the mediators. The initial mediators that are released are interleukin 1 beta, interleukin 6 and tumor necrosis factor. I'll be repeating these mediators again and again so that you kind of remember them. Anytime they flash in front of your eyes, you will come to know where and when they acted. So you don't have to mug that up. So in the initial first 12 to 24 hours, these initial mediators are released. So what happens? The osteoblast, which was lying very dormant, but it had those receptors, the TNF receptor also and the IL-1 beta receptor also. So once these mediators are released, they are taken up by these receptors. If the nuclear factor ligand, the rank ligand or the TNF receptor or the TNF-R1 gets activated, it leads to osteoclastogenesis. How? There is an osteoclast in its vicinity. This receptor is present on the osteoblast, the TNF receptor, or you can also call it rank ligand, which is receptor activator nuclear kappa ligand. It is present on the osteoblast. Once it comes in close vicinity with the osteoclast, which is having that rank on its surface, it's going to bind. If this rankel binding occurs, only then the osteoclastogenesis is going to happen. So please mark my words. Rank is receptor activator nuclear kappa ligand rank L. If it binds with the rank on the osteoclast, then the osteoclastogenesis happens. But there is also a catch here. You see, I have made, just for my understanding, I have made these as triangular receptors because they are similar in shape. If you have that triangular cap on the osteoblast, it is going to fit into the triangle which is present in the osteoclast. Then that binding is going to happen. But there's another receptor which is an osteoprotegrin, which is present on the osteoclast. This is also triangular in shape. So now when rank-L comes, it doesn't know where I should bind. Whether I should bind on rank or I should bind on OPG, that is osteoprotegrin. If it binds with OPG, no osteoclastogenesis is going to happen. So it is important that rank-L binding occurs for osteoclastogenesis. Now you can say that what was IL-1 beta doing then? Interleukin 1 beta is also very helpful in it because if it is present in good quantity, it will help the rank-rankle to bind, and then the osteoclastogenesis is going to happen. Another receptor antagonist, which is interleukin 1 beta receptor antagonist, also plays its role here. If that receptor antagonist becomes more, it will not let the interleukin 1 beta receptor to act. So again, please mark my words, there is an activity ratio by the name of interleukin 1 beta divided by interleukin 1 beta RA, which is receptor antagonist. So you can see, if the receptor antagonist becomes more, osteoclastogenesis becomes less. So activity index becomes less. But if interleukin 1 beta becomes more, activity index becomes more. So osteoclastogenesis happens. So two major things which are important are rank-rankle OPG ratio and interleukin 1 beta oblique interleukin 1 beta receptor antagonist ratio, which is activity index. Please mark my words. This happens initially in first 12 to 24 hours. After 7 to 10 days, what happens is then the reparation process is going to begin, and all this is going to get reversed. Like the OPG ratio is going to become more, the interleukin 1 beta receptor antagonist is going to become more, there is going to be more growth. So all the growth factors like the TGF beta, which is the transforming growth factor, or the insulin growth factor, or the fibroblast growth factors, all these are going to become more. The vascular permeability is going to get decreased. So all these are going to become repaired in 7 to 10 days. If you remember, our 3 phases of tooth movement were initial phase, lag phase and log phase. So this is basically the lag phase. What happens in the pedontal ligament? Initially we have said that the vascular permeability increases and all this effect is taken up by the blood vessels which form the precapillary network. But then after that, after the initial phase, the pro-inflammatory cytokines, we know it's going to increase, and I've told you what effect it can have on the alveolar bone, the blood cells and the fibroblasts. Besides these cytokines, these are cytokines, interleukin 1 beta also, TNF also, TNF alpha also, tumor necrosis factor. Besides this, you have enzymes, which are lactate dehydrogenase enzymes, other enzymes also like aspartate transaminase or TRAP. You should also remember a few words like TRAP or aspartate transaminase, which is AST, or chemokines. Chemokines are basically which are going to dissolve the extracellular network. These are enzymes which will act on the extracellular networks. They are collagenases, fibrogenolyses. All these are like MMPs, matrix metalloproteinases. I will not go into too much of detail because you will not be able to remember that much. But you just remember, alveolar bone is very important, how osteoclastogenesis happens, rank-rankle ratio, the interleukin 1 beta over interleukin 1 RA, which is receptor antagonist ratio, which is very important. Rank-rankle OPG is also very important. Besides that, few other enzymes are important like lactate dehydrogenase, TRAP, aspartate transaminase, which are going to cause resorption. Apart from that, you have alkaline phosphatase, which are enzymes which will cause bone deposition. Apart from that, you have matrix metalloproteinases, which are a form of chemokines. As I said, that the first brunt where it was taken at the cushion, at the extracellular surface, which has collagen. So if you have to dissolve the collagenases have to be there. So they are MMPs, which is matrix metalloproteinases. Besides that, it does not only affect the surrounding structures, it also affects the structures which are within the tooth, like the pulp. So how it happens? Again, those mediators have already been released. The interleukin 1 beta, interleukin 6, TNF alpha from the pulp fibroblast, they have been released. So when the neurogenic inflammation is going to happen within the pulp, the pain receptors are going to get activated. So neuropeptides are released. That also you should remember, substance P that gets released, which is giving you that pain perception. So initially when you see the first appointment of any orthodontic patient, the patient always have pain, will complain of pain within 1 to 2 days only. So that releases very initially substance P, and it also stimulates the rancle and pulp fibroblast and all. You don't need to remember that much, but the growth factors, if they start getting released, then they will kind of revert the action of this rankle in the pulp. So again, there is a feedback mechanism which happens. When you talk about the gingival tissue, that also takes its brunt, and the nitrous oxide synthesis, which is important mediator in the gingival tissue. In the initial phase, it forms these two enzymes, and they can cause both resorption and deposition. There is another second type. When we talk about these are the primary messengers, okay? But these also form the prostaglandins, which is prostaglandin E1 and E2, PGE2 and PGE1, which are most important. This also has a dual action. Basically, on the surface, it is going to cause apposition, and otherwise, from the endothelium, if it goes from the bone, from the blood vessels, then it is going to cause resorption. So prostaglandin E2 is also very, very important as a mediator. You know, it has been tested. They have kind of injected prostaglandin E2. There is also one thesis that went on in SDM Dharwad here in our country, and the particular researcher had to take a lot of ethical clearance, had to take a lot of problems for the ethical clearance, because he wanted to inject it in himself and see what happens. He was already undergoing the orthodontic treatment. So definitely, if you kind of inject it, then the resorption, it definitely happens, and the tooth movement becomes faster. That's what they have shown. But yes, there are animal studies proving that. This you basically remember because many times they ask you what happens when corticosteroids are given in the initial orthodontic treatment or the NSAIDs. Are they preferable to give? No, you should not give them, because it will hamper the prostaglandin synthesis, and hence the bone resorption will not occur and the tooth movement will not occur. So once we formulated a basic concept, now let's go to what is orthodontic force and what is orthopedic force. When we talk about what is orthodontic force, it is basically again 20 to 150 gram per tooth when you apply, and for the purpose of affecting tooth movement. But if you increase that force to above 300 gram, which is not only sufficient for the tooth surface, but it can move the bones, the skeletal structures. So for that, you deliver it for 12 to 16 hours, and you have various orthopedic appliances like headgears or face masks, or you can give any kind of appliances for that. What is an optimal force? When you talk about optimal force, definitely optimal force will be you apply any force and nothing happens. I mean, you don't occlude any kind of blood vessels, you don't formulate any kind of mediators. Well, that does not happen. But the orthodontic force ideal would be if you equate it with the capillary pressure, bed blood pressure. Because you see that initial effect was taken up by the blood vessels only when they form the precapillary network. So if you kind of maintain that pressure to the capillary bed pressure, then no harm is going to happen. You will not have any root resorption, you will not have any undue undermining resorption. So that pressure is equal to 20 to 25 grams per centimeter square of root surface. The current concept is also that whenever you apply this orthodontic force, it will be taken up as an external mechanic stimulus, and it will restore its equilibrium by remodeling the periodontal supporting structures, but will have no deleterious effect. That will be an optimal force. For every kind of tooth movement, the optimal force is given. For tipping, it is 35 to 60. For bodily, it is 75 to 120. Root uprighting, 50 to 100. Rotation, 35 to 60. Extrusion, 35 to 60. And intrusion is 10 to 20. So there should be no trauma, and there should be no round tripping. There should be no resorption. But the light forces only are preferable. The heavy forces, as you all know, can cause hyalinization. Now you can very well tell me that when you apply heavy forces, what will happen to the extracellular matrix, what will happen to the precapillary network, and what will happen to the alveolar bone. So you know it very well. Now we've discussed it in the previous slides. Let's talk about some basic theories of tooth movement. They are pressure tension, bone bending, and piezoelectricity. So when you talk about pressure tension, is it that simple that you just apply pressure and on one side the pressure compression happens, and the other side the tension happens? The root is also not a very straight kind of a structure. It has its own micro irregularities. So if you see microscopically and histologically, you will see that they have micro areas of compression and tension. But overall, when we consider, if you take this example of a single rooted tooth, what's going to happen? If you try to move this tooth lingually, you see there is a negative marking there. The negative marking shows that in the area of compression, the bone desorption is going to happen, and the deposition is going to happen where you have the positive sign. This is opposite to what we are going to learn in the electrical stimulus. So you have the negative here where the compression is going to happen, and on the opposite surface the bone deposition is going to happen. So it kinds of shifts the tooth, and it also keeps on forming the bone alongside. You know what all mediators are involved, that all are going to have their role in this kind of remodeling process. So if you have a frontal desorption, there are three phases basically. So when you kind of put the initial pressure, if it's not too ideal but a normal kind of a force, you have a light force, then what happens is that the osteoclasts are going to line on the periodontal ligament surface. When you have a periodontal ligament, then you have an alveolar bone. On that surface, when the osteoclasts are going to line, there has to be a frontal desorption. But on the contrary, if you have a very heavy force, so what's going to happen? The tooth is directly going to touch to the alveolar bone. There is no space for the blood vessels to line there. There is no space for the osteoclast there, because it requires that blood supply, it requires that form of mediators. So then there has to be a cell-free zone which is formed, because everything is going to get occluded there. You can also see histologically as pycnotic nuclei or acellularity. But then this kind of acellular mass is going to hamper the tooth movement. It's going to stop the tooth movement. So what happens? This resolution of this mass is going to occur by the macrophages or the foreign body giant cells which are going to occur, or the osteoclasts. From where will they occur? Where will they come? Because we have occluded this pathway. So it is going to come from the other pathway, from the undamaged area, which is the marrow spaces. If they come from the underside, then it is called the undermining desorption to clear the hyalinized zone. Now you understand what is the difference between a frontal and an undermining desorption. A frontal desorption will happen when you apply a normal force and the tooth will slightly move in the periodontal ligament space, and it is going to line there, and the osteoclasts are going to line that surface towards the alveolar bone, and the resorption is going to happen. But in the areas where hyalinization happens due to very heavy force, the occlusion of the blood vessels takes place, and there is no way that the osteoclast can be viable in a frontal desorption area, then this resolution is going to happen from the underside, from the marrow spaces. From there, the macrophages and the osteoclasts are going to come and going to eat away that hyalinized area. It's going to again become free, and then the log phase is going to happen. Okay, then the tooth movement is going to happen. So these are the basic phases. Pressure tension, well, hyalinization will also depend. It's not only the amount of force that we are going to put. As I said, that root is not a straight line surface. It has micro irregularities. So it will also have its own zones of hyalinization and zones of normal force. It depends on the shape of the root. The short root, size of the root is also important. Short root is definitely going to be more prone to the hyalinization. Besides that, you have type of tooth movement. Tipping definitely has more areas. Translation does not have that much of area of hyalinization. The direction of force, duration, distance, and continuity. If you talk about direction, Well, in the direction where the force is happening, there definitely the compression area that is more prone to hyalinization. Duration, if you keep on putting that heavy force, then the hyalinization zone is going to become more and more. And it's not going to get chance to dissolve itself. Distance, if you move it to a shorter distance and you apply heavy force, then definitely the hyalinization is going to happen. Continuity, again, the continuity of the force that you apply. Patient related factors, greater age when you have more calcification and you don't have that much of leeway in a periodontal ligament space, you don't have that viability of the blood vessels, then definitely the hyalinization zone is going to become more. Individual tissue reaction definitely can be also influenced by some kind of medication if you're taking, or some kind of osteoporosis that you're having, or endocrine imbalance you're going to have, that also. Forces of occlusion also play a role, but now the new formed structures like the supraalveolar structures, they also play a role, because these transeptal fibers, they connect one tooth to the other tooth. So if you see that it's not only one tooth that's getting affected, but it's also transmitting this effect to the surrounding structures. Well, as we talked about in the first slide of Mustafa model, we said that this pressure tension is not a secluded phenomenon. It works in unison. Now, when you try to put some pressure, what's going to happen? We are not only talking about the frontal desorption, the undermining desorption. It's not only the periodontal ligament space that we are bothered about, but what happens um, to the bone structures, to these tooth structures, solid structures? They bend. They bend under this stress, and these stress lines can be seen throughout the bone. So you see, in this particular single rooted tooth structure, as you see on the left side, there's an arrow where it is indicating that the force is being put from the lingual side. So what is happening on the buccal side? The compression area is formed. On the lingual side, a tension area is formed. When the tension area is formed, the periodontal ligament fibers, they stretch. When they stretch, they will pull the alveolar bone surface with them, and there will be a concavity which will be formed, because it will be thinning of that alveolar bone on the lingual side. While on the buccal side, there will be a pileup of that periodontal ligament structures, so you will see a convexity. But the negative structure here indicates, the negative sign here indicates the formation, bone formation, and the positive structure here is going to indicate the desorption. So this is only which is indicated here. The concave side, as I said, when you stretch the periodontal ligament, they're going to stretch the alveolar bone also with them, and the concavity is going to form. And that is electronegativity. It is causing bone formation. But on the convex surface, on the compression side, you see, there is a pileup of the periodontal ligament surface. So it is going to form the osteoclastic activity. It is going to form the osteoclastic activity. The Wolff law then definitely explains that there is a relative slowness of en masse tooth movement. It is going to explain these facts, because uh, when these connectivity happens, then there is a relative slowness of en masse tooth movement, and bone flexion is needed for that rapidity of alignment. The rapidity of tooth movement also towards the extraction side, and relative rapidity of tooth movement in children who have less heavy calcified and more flexible bones. We've already talked about all these factors. Well, there is also something called a regional acceleratory phenomenon. When there is a stretching of the alveolar bone, as I said, on the tension surface, and there is a there's a concavity which is formed because of the thinning of the alveolar bone, then there are various kinds of cells and mediators which are going to be attracted towards the site, and that is a regional acceleratory phenomenon, which is also called the bone formative phenomenon. Apposition of bone takes place here. But in the complex zone, where the convexity is there the bone desorption is going to happen. Bioelectric theory definitely favors piezoelectricity as well as streaming potentials. And when you talk about piezoelectricity, it is basically a phenomenon for many crystalline structures. And these crystalline structures can be seen in collagen. These crystalline structures can be in hydroxyapatite, but they are also seen in the organic structures like collagen and collagen hydroxyapatite surface, mucopolysaccharides. All these also are going to show these electric surfaces, flow of electric current. When you put a force on one kind of a crystal lattice, the electrons are going to go from one area to the other area, right? And they have a property of quick decay, because as soon as you remove the force, the electrons are going to flow back. So that is a reversal. And as soon as you put that force, it will initially spike and then it will stop. So there is a decay which is very fast. But this does not happen. If you put the force on any tooth, you see that this piezoelectricity does not have that much of role, because the force still is happening on the tooth. Even if you remove that force, even if you remove that force, it is not going to go back, right? So there is no reversal phenomenon also. So apart from the inorganic structures, the organic crystals also show piezoelectricity, and they are seen in the stress tissues. What can basically explain this fact is a streaming potential, because these crystals are not present on their own. They are bathed by the periodontal ligament fluid, the fluids which are present there. So the electron that are flowing from one area to the other in the crystal lattice are also acting with the electrical potential which is being generated in these fluid areas. So they don't decay that soon. It does not have a quick decay rate. It does not have any reversal phenomenon here. So this is more applicable because it is in that fluid structure. It has that conduction and the convection mechanisms. So once we've talked about the all kind of theories that are applicable to the tooth movement, we also should mention what are the types of orthodontic forces. So you have some kind of a continuous force, which is when I put the fixed orthodontic braces, and along with that I put a night-eye coil spring. I put certain springs which will deliver a kind of a continuous force to the tooth. Does that continuous force means that they will have equal amount of force for a long span of time? No, you will see that there is a certain decay which you see when there is a decay, and then again there is a reactivation, then it reaches the force level, then again there is a decay, but the force level will never reach zero between the appointments. Okay, it is not going to reach zero. On the other hand, if you talk about interrupted and intermittent, they are not clear cut, they sometimes overlap also. Because when you talk about interrupted, suppose I've put a screw, a fixed high rack screw, okay, or any kind of screw like a distraction screw. So if I put that screw, if I activate it, it will only be active till the time there is activation in the screw. After that, the force is going to become zero. Then next time when the patient is going to come to you, you're again going to activate it, then again it's going to get activated, and then it falls to zero. On the other hand, if you have a removable appliance and you are putting that appliance, some force is being generated, the force level even in a removable appliance is going to go low. If the patient keeps on wearing it, the force is going to go low, but it never reaches zero. But if the patient removes it and keeps it and then puts it back again that same force is being applied. So you see, it does not fall zero here, but definitely there is an interruption. There is an intermittent force which is being applied when the patient is going to remove it and then again place it back again. Even in continuous forces, now that we've understood the concept of heavy forces and the light forces, so you can very well imagine if we have a heavy continuous force, what is happening? There is going to be complete occlusion of the blood vessels and the hyalinized zone is being formed, and you keep on applying that force, then there will be no way that hyalinized zone is going to get resolved. So always a light continuous force is more desirable. If you have to put a heavy force, then it has to be a heavy intermittent force, because ultimately it is going to become zero. In that period where you see it becomes zero, again it will still have time to recover, to resolve itself. So before putting that heavy force again, the resolution will have taken place. So if you apply heavy force, let it be intermittent, and if you have to apply a continuous force, it has to be a low continuous force, which is most preferable. Then it all boils down to genes. Well, these are few genes that you have to kind of, you have to know. We've talked a lot about them, but apart from that, you also have some genes like bone morphogenetic proteins, BMPs, transforming growth factors, insulin growth factors. You remember I talked about them when we talked about the formative enzymes, okay, or the formative growth factors. You have homeobox proteins also, which are leading to osteoblast differentiation, MSX1 for bone development and modeling, bone patterning, skeletal adaptation, MSX2, CBFA1, which is earliest and most specific of bone formation, HOXA2, which is again which is opposing the CBFA1 expression. For osteoclastic, you have OPG. Cathepsin K is again, which is also going to dissolve the extracellular matrix. Chloride channel, which is also important, CLCN7. You just need to remember a few. You can just recall them if any question comes related to it. This is a table which I have taken from standardized books, and I've also mentioned those books as reference later on. So if you want, you can just go through it. We have kind of covered almost every mediator which is mentioned here. We have covered interleukins, we have covered osteoprotegrins, we've covered prostaglandins, we've covered RANKL, we've covered NFKB, and also some metalloproteinases where tissue inhibitors are present. But there are these biological procedures which are also being taken to our benefit for acceleration of tooth movement, this tissue injury. These procedures, just remember, they are corticotomies where you just you kind of give an incision, and then you make a segment of a particular tooth, and then again this regional axillary phenomenon is going to happen, and then the bone formation, it is also going to cause the acceleration of tooth movement. Piezo small kind of marks that you're going to give to cause that regional axillary phenomenon. Micro-osteoperforation, again the same thing, where you're going to give the you're going to perforate the bone with the very small incisions. Orthodontics, again, where you make a segment of each tooth. So these are all acceleratory phenomena. The procedures based on physical effects are accelerant, which is based on vibration, and biolux, that is infrared light, again that is going to cause the acceleration. And again this therapeutic ultrasound is coming. I don't know how effective it is, but if any question comes, you should know that these are the procedures that are being used. These are the references, and I'm sure you have not read these books till now, but if you get time and if you plan to do a PG in orthodontics, few standardized books you have to know, and these are very good books, and many of my concepts have come from these books, so I really recommend these books. So orchestration of biology of tooth movement is still not complete, it's still lacking, but here is a small attempt that I have made. I hope that you've understood a few concepts, and a few mediators definitely will come at the top of your mind when you see any question and you read about it anywhere. So that's my whole idea, that if I can motivate you to just understand and just answer the question not blindly, just understanding the concept is more important. So thank you so much.